A warm good morning to one and all. Now let's begin the online sessions for the invited talks of the two-day national seminar on interdisciplinary research approaches and Research Scholars Meet 2022. First of all, let me take this opportunity to sorry Dr. Aravind Kumar for the short delay in beginning this session. Today we have Dr. Aravind Kumar Rengan, Associate Professor, Department of Biomedical Engineering, Indian Institute of Technology, Hyderabad. A medical doctor with intense interest in research in cancer nanomedicine. After completing his MBBS, he did Masters in Nanomedical Sciences at Amrita College of Nanosciences, DST Center for Excellence, and PhD in IIT Bombay. Currently working in the area of photothermal therapy for cancer using multifunctional nanoparticles. He has won the Young Scientist BRICS Young Scientist Forum Award in 2021, currently has 3 patents and nearly 50 publications. Today, we have also Dr. Sister Alfonsa Matthew, Department of Mathematics, St. Thomas College, Thrissur, to chair this event. I welcome one and all to this event and request Dr. Aravind Kumar Rengan to deliver his talk.
Thank you, madam. I will get started with my presentation.
Hello, I request Sister Alfonsa Matthew to chair the session and begin this talk.
Okay, good morning, all of you. Thank you, Anandor, present this virtual platform. Actually, I feel honored to chair this important session and to introduce, just like we have already heard about Dr. Aravind. So I don't think we need any introduction. So I think now it's the time we can listen Dr. Aravind Kumar. On behalf of St. Thomas College and everyone present here, I welcome Dr. Aravind Kumar to speak to the audience. Over to you, sir. Thank you.
Thank you, sister. Thanks a lot for the wonderful introduction, and apologies for the slight glitches that I had. So let me get started with today's talk. Today I will be speaking about mainly my experiences on the art of writing research articles and proposals. So I don't want it to be a cliche wherein I present all the necessary components that gets involved in research articles and proposals. Rather, I decided like I will speak about what are all the mistakes that I made, and so that people can learn from my experiences. Let me get started. I will call myself as an accidental researcher because I was forced into medical school. Coming from a middle class family, you have only two options, either to take medicine or engineering. And because my sister did not get into medicine and I got a government seat, my parents advised me and sort of forced me into medical school. But my interest was in engineering. So after I completed my bachelor's in medicine and surgery, I wanted to do something that is related to engineering, and nanotechnology was coming up in a big way. So I studied nanotechnology along with engineers, chemists, physicists, and that is where I met Dr. Jones Naduvat. He has been my batchmate for nearly 7 years. 2 years of MTech program at Amrita Center for Nanoscience, followed by 5 years of PhD program at IIT Bombay. And that is where we exchanged a lot of ideas. And because I moved with a number of people from different backgrounds, I got to know many things that other researchers in the research field would have missed. And today I will share with you all those experiences that I had in the past few years. So I did PhD in an engineering institute, and by the time I completed the PhD, it almost took 15 years after my plus two. Plus two, I completed it in 2001, and when I completed my PhD, it was 2015. So you can imagine the amount of experiences that one would have had after their schooling. And now currently I'm working as a faculty in biomedical engineering department. I'm teaching medical subjects to engineering students. Yeah. So coming to this topic, The topic given to me by Jones was to speak about the art of writing research articles and proposals. And both this research article and proposal is something to do with science. So we can cut short this topic into art of writing science. Okay. Now, if you consider what is the difference between art and science, art is a kind of a skill, and it may not be repeatable. Today, I will only give you some suggestions which can help you to write papers and to formulate ideas, to write proposals in a better manner. But what works for me may not work for everybody. At the same time, whatever I say is not the all and end of it. There are many more ways to improve it. And science, of course, should be repeatable. That is why we have materials and methods in each and every science article that we write. Okay. There is a disclaimer that I want to make. Just because I am going to speak about research article writing and proposal writing doesn't mean that I am authority in it. I am also a researcher who is in the learning process. Okay. And just to give you an example, after I entered into the research arena in the year 2008, where I started my master's in technology program, that is where I was introduced to research. Generally, medical students in India have very little exposure to research during their bachelor's program. We don't have to do any project work. We are mostly exposed to the clinical work. So I got exposed to research work only when I entered master's program at Amrita Center. And after I entered, I did some research work for nearly 2 years there during my master's. And then I went on to IIT Bombay and then started continuing my research work. But then it took nearly 7 years to publish my first research article. So only in 2014 December, my first research article was out. Okay. But when I started my research work, I was a bachelor. By the time my first research article got published, I already had become a father, and I got married, and I already became a father. So that is the generation of what is that years it took for me to publish my first research article. And all these things definitely tell you that I may not know what to do, but definitely I know what not to do. Okay, so I will be sharing my experiences wherein I will point out all the mistakes that I made, so that you need not repeat those mistakes whenever you venture into writing proposals or writing a big research article. Coming to research, why do we do research? Okay, if you ask this philosophical question, obviously the answer is you do research for the betterment of humanity. But there is a very important pertinent point to it. I'd like to quote the words of John B. Goodenough. Okay, he is a Nobel laureate. He got Nobel Prize in the year 2019 in chemistry for what is that invention of lithium-ion batteries. Okay, now we use this lithium-ion batteries in various gadgets. Okay, cell phone and everything. Okay. And when he was called and when he was informed that he has got Nobel Prize at the time in 2019, he was 97 years old. Now he is 99 years old. So he is one of the oldest living Nobel laureate. So he laughed and he said, they asked him, how do you feel? He said, like, at this age, I have nothing much of an expectation, and I do not expect that I will be given Nobel Prize. And he said a very important statement: all our inventions are morally neutral. So I want to extrapolate it. The first invention of the human civilization is the wheel. Okay, that is why we say, like, when somebody tries to redo some work, we say that don't reinvent the wheel. Okay, so wheel is considered as the first invention of man. But if you look at what are all the consequences, not everything has been very comfortable, or not everything has been used for the betterment of humanity. Because wheels were invented, then we started moving very fast. Okay, now we have so many vehicles, we have a lot of air pollution, we have water pollution, we have traffic jams. Okay, and everybody knows that a lot of online sessions are happening today because of the pandemic. Okay, in the year 1919, that is exactly 100 years ago, we had the Spanish flu. But Spanish flu did not travel all around the world. Okay, it was limited to a certain region, and I'm not very sure if India had Spanish flu. The reason was at the time the air travel was not that frequent. Okay, but we have enjoyed the benefits of air travel. But today, because of air travel only, COVID was spread to all the countries. Okay, so philosophically, all the inventions that are happening are morally neutral, but we should always venture into asking questions which are able to make our life better in the current scenario. So understand the lacunae or the unmet need that is currently present. Okay, that lacunae could be addressed either directly or indirectly, and this is the central point of any research topic. Okay, is it addressing lacunae? Is it addressing an unmet need? Okay, and as long as your lacunae or the unmet need is very strong, there are high chances that the proposal gets funded or the research paper, article, or research work gets published. Now, coming to the other technology that is very, very important in research is, apart from addressing the lacunae or meeting the unmet need, we also have to look at how novel the work is. Okay, what is new in your work? Okay, is it incremental? Is it substantial? Or is it disruptive? Okay, when I say disruptive, it is something that will cause a paradigm shift in the way how things work. Okay, a simple example is in electronics and telecommunication, cell phone and the memory gadgets, memory sticks that we use. They are all considered as completely disruptive technology. It caused a paradigm shift in the way how we things work and how we communicate faster with each other. Generally, it is said that substantial work, okay, or sometimes even disruptive work gets into high impact publications. Okay, if we do just incremental work, we can never achieve a high impact publication. Okay, what is high impact publication? We'll get to back to it later in my oncoming slides. Now, when it comes to novelty, we have various ways of expressing novelty. One is a straightforward way. We say that if we do this, this is considered as novel because not anyone has attempted it, and it has many merits of doing things in that particular way. The other is conceptual re-engineering. Okay, I'll speak more about conceptual re-engineering in my oncoming slides. And the other thing that we have to pay attention to is, in research, we have two types of entities. One is convincing evidences, the other is facts. Okay, so when a researcher, a student researcher who ventures into research, one thing he or she has to keep in mind is that whatever that is given in a research article may not be true. Okay, they are convincing evidences. Okay, they are not facts. Okay, facts you will only get in books. Okay, where lot of research work gets into literature, and people understand this after certain time period of time, and then they consolidate it, and finally books are written with those research papers which are very strongly suggestive of these evidences. Okay, so there is a difference between facts and convincing evidences. I'll give you one example from my research work that is related to research work. One is we use a material called as cetyl trimethyl ammonium bromide, or CTAB in short, to coat nanosystems or to synthesize nano rods. But earlier, people found a lot of toxicity with this CTAB. And the earlier thought process was it is because of the highly positive charge that CTAB has that leads to toxicity. Later on, people found that it is not because of the charge, it is because of the material per se that CTAB is toxic. Okay, but you will find articles not only for the charge leading to toxicity, you will also find articles for the material per se leading to toxicity. So the more the convincing evidences that you gather, the better you understand about your research. Okay, and when you venture into a particular topic, you need not have to blindly accept the convincing evidences that are given in one particular paper. You will have to read a lot of paper, and then you can convince yourself, okay, these are the things that are given in one particular school of thought, and these are the things that are belonging to another school of thought, and it is up to you to accept what type of school of thought. I'll give you certain examples from my research work. My research work mainly is dealing with photothermal therapy, wherein we synthesize certain nanoparticles. We use light to activate these nanoparticles. This nanoparticle does convert this light into heat, and that heat will kill cells. And we rely on targeting factors like active and passive targeting, so that these nanoparticles reach the tumor cells. And we have synthesized various types of nanoparticles like nanorods, nano cages, nano shells. Okay, and I will just run few slides, and then I'll get back to the understanding of reverse engineering concepts and other things. Okay, so our earlier idea was to take these nanoparticles, put them inside delivery systems which are called as liposomes, and then we use it for drug delivery or for photothermal therapy, wherein heat is used to kill the cells. Now, this is a typical example of how photothermal therapy works. I will not be going too technical. You can see that when there is no treatment, or when even light is showered, there is laser light, or when particles are incubated with cells, these are all, whatever you see in green, are all cancer cells. Okay. If they are alive, you see a lot of green cells. If they are dead, then you won't get any green signal. And you see that when we combine these two things together, that is nano cages with laser light, you see all the cells becoming dead. So this is what is considered as photothermal effect. Okay. So as a part of my literature survey, we found that there are only a few materials which has gone into clinical trials. One example is silica gold nanoparticle, which has entered into clinical trial for human use. But they have used it only for therapeutic purpose. They did not use it for diagnostic purpose, though gold is a very good example for diagnostic agent. It has very good X-ray contrast. It has very good optical contrast. But then the material stays in the body for a very long time, as a result of which you cannot use it for diagnostics. So we wanted to use a material which is degradable and disintegrable, so that it gets cleared from the body very fast, so that we could use it for both diagnostics and therapeutics. So this was the material that was designed by us. So first we synthesized liposomes, which are carrier systems, and we coated that with gold seeds, and this has a kind of co-shell effect, and it also kills the cells through photothermal therapy. At the same time, it disintegrates to small particles of gold, which are capable of getting cleared from the body very fast. Okay, and here I want you to focus on these signals that we got from our sample. This is the lipos sample, from which we get very good signal, and this is the commercially available iodoform. This iodoform is used for X-ray contrast, whereas the lipos nanoparticles were synthesized by us mainly for therapeutic purpose. Okay, but apart from therapy, this is also capable of giving signal which can be used for diagnostic purpose. So in a way, we can use these particles for therapy as well as for diagnosis. Okay, so we did few more studies wherein we injected this particle, and we found that they are capable of getting cleared through the renal route. So here you see B stands for bladder region, where you get signal from the bladder. Whereas when we use control, there is no signal coming from the bladder region, indicating that this material is capable of undergoing very good clearance from the body. So the idea of this paper was to show to the world that we can use such materials, that is lipos nanoparticles, which can after the job of killing cancer cells is done, they can disintegrate and then come out through urine. Okay, and this was the first known nanomaterial that is capable of undergoing renal clearance. Before this, that is before 2015, before this paper got published, there was no other nanomaterial which is shown to have good renal clearance. Okay, so that was the novelty in this work. that we created a material which is capable of getting disintegrated in the body, and it is capable of achieving very small size which can get cleared through the renal route, apart from photothermal therapy. And this led to a new branch of study wherein people started working on biodegradable nanosystems for photothermal therapy, and this paper has been cited more than 300 times as of now. Then we started working on certain other materials like fluorosomes, which are like lipid substances taken from spinach leaves, okay, through extraction method. And we started working on completely degradable substances like liposome with bioactive molecules. Here we have used a bioactive molecule from Anthocephalus cadamba, which is known to kill specifically cancer cells, and therein we can achieve specificity. And these kind of completely degradable lipoCFAC molecules were used for creating autophagy, which is a form of cell death. I'm not going into the technical details. I will just show you a few experiments, and then I'll get back to what role it plays in writing a research article. So here we have added, in addition to the liposome nanoparticle, we have added curcumin, which is a medicinally known plant extract, and we have used it for adjuvant therapy, which can help in better treatment of cancer cells. So here you have a group which is having only liposome with laser treated group, that is only photothermal, whereas here you have photothermal therapy combined with curcumin delivery, which is having better effect. Here whatever blue colonies that you see, these are all cancer cell colonies, whereas here you don't see any colonies, indicating that this kind of therapy is better than other form of therapy. Now, coming back to whatever I have shown to you earlier, this I will call it as observation slash conceptual reverse engineering. Let me go back to the slides once again, and I'll tell you this is not as straightforward as how it is written in the research article. I'll tell you from this slide, okay? So this is what the paper says, that our approach is to create a nanoparticle coated with gold so that it can disintegrate, and it can, after the job of photothermal therapy mediated cell death is completed, it will disintegrate and will come out from the body, okay? But in the real world, or in practically speaking, you will not encounter such things. So I will tell you what happened actually, okay? So I started my research work keeping silica gold as an example template, and we wanted to create a material which is as good as silica gold, okay? And I had two guides, and one guide was working on gold mediated material, and another guide was working on liposome mediated material. So we combined that together. So instead of silica, we used liposome, and then we coated that with gold, okay? But unfortunately, every time when I hit that with laser, the material used to degrade and it used to get disintegrated. So we were not getting consistent results. So the first time when I do this photothermal therapy, the results were good. But subsequently, when I keep doing photothermal therapy on the same material, we were not getting good results. That reason is because this was degrading, okay? But in reality, we have developed an unstable material which is not that great when compared with silica gold. But if you look carefully, there is always an opportunity in these kind of situations wherein you can call this unstable material as a degradable material, and then bring out the disadvantages of silica gold and bring out the advantages of this degradable material, and then project those things, okay? And then you also have to look at what are all the other good things of using such materials. One thing I told you, it can be used for diagnostic purpose. Not that silica gold cannot be used, silica gold also can be used, but the problem with silica gold is that it is going to stay in the body for a longer time. And for any clinically viable diagnostic agent, it has to come out of the body very quickly, because a doctor, when he is subjecting the patients for diagnostic purpose, he will also subject the patient for prognostic imaging. That is, after the treatment is given, whether the treatment is working or not. And if the material which is going to give the contrast is going to stay in the body for a longer time, then it is going to become an artifact, and it will confuse the doctor whether the signal is coming because of the disease or because of the remnant particle that is present. So that is why we want to have a nanoparticle which is completely degradable or disintegrable and which can come out of the body very fast. Okay, so these are the opportunities that we have in this unstable material. So we never use the term unstable, though that was the actual material that we had prepared. And here again, coming to this particular research where we have created lipid from the spinach, this was again not a straightforward research. There was a time period where our lab was falling short of funds, and so we could not buy these costly lipids. Hence, we have to look at alternates for these lipids. So one of the alternate that we got was this spinach leaves, which was a little rich in all the phospholipids, and so we extracted what is that lipid from it, and then we created liposomes along with polydopamine. But this is not the right way to project a paper. Okay, what is the good thing available in this is like it is capable of giving a fluorescent lipid. Okay, because whenever we extract, we also extract the chlorophyll molecule along with it, and chlorophyll is a well-known fluorescent agent. And you get the benefit of not only the lipid, but also the fluorescence that comes out of chlorophyll, and we can use it for therapy as well. We call it as photodynamic therapy. And here we have used this plant extract molecule. Again, owing to shortage of funds, we had to rely on certain affordable materials, and that is the reason why we went for this kind of research. And when we write this paper, we project what are all the good things that are available with our material. So in this case, we typically ask 3 to 4 questions. Is it the material affordable? Is it degradable? Can it cause selective drug delivery? And can it reduce the gap between identification and treatment? So in cancer, when a patient gets identified, especially those who are below poverty line, they'll have to wait for days to months together before they are getting their actual treatment. Okay, because in our country there is private setup as well as government setup, and private setup it is very costly. Not everybody can afford for it, and in the government setup there is a lot of waiting time involved between diagnosis, that is identifying a cancer, and then getting it treated. And this waiting period, if it is a little larger, then the cancer may also spread and go to advanced stage where it becomes untreatable. So here we have used a material which can not only use for treatment, but it can also be used for diagnostic purpose. That is, it is giving signal. So we can in a way reduce the gap between identification treatment. The moment we see it, we can immediately use it for an X-ray or laser trigger, and we can treat the tumor then and there. So these are the questions that we ask based on the results that we get. And when it comes to writing a paper, you will have to project these critical challenges and then have a flow. Okay, but in the real world scenario, this is not the way how things occur. Okay, that's why I say that this could be called as a conceptual reverse engineering. Okay, based on the results that you get, you identify what is essential and in what way this is fitting the critical challenges that are there, in this case as far as cancer treatment, and then project those questions, and then come back and say that these are the points why this material could be considered as a better one for addressing all these critical challenges. And as I said, observation is also very important. Okay, coming back to another thing wherein targeting was involved. This is what is a typical way by which we use nanoparticles to passively target tumor cells. I'll keep it a little simple. In the tumor cell, the vasculature is going to be very leaky, so that these particles go and accumulate in the tumor region, and the drainage system is not that developed. So as a result of which, it will stay there for a longer time. In that way, we can use these nanoparticles to target cancer tissue. Now, some of the materials that we used, they did not have good targeting ability. And some other material, say for example, in this case, instead of lipose AU, we had used PLGA AU. And PLGA is a polymer, it's an FDA approved polymer, and that PLGA AU was able to accumulate in the tumor region. Here you see this is a dye control, which when we inject it, it goes everywhere and gets cleared from the body. But when we use a nano system made up of PLGA coated with gold, it is able to reach the tumor site on its own. So here there are two challenges that are addressed here. One is this does not have any active targeting moiety. So active targeting moiety, what I mean is something that will specifically take the nanoparticles to that particular area where the corresponding receptors are. But those active targeting molecules are very costly. So here, without the need for active targeting molecule, these particles are able to home into the tumor site. So that is what we call as passive targeting. And the advantage of passive targeting is that it can be used for many other types of solid tumors as well, because there is no specific markers present here. And at the same time, it is very much affordable because we don't use any costly active targeting molecule. At the same time, you can use it for diagnostic purpose. You can inject this, and if there is some signal coming from the region, you can to an extent decipher that this is because of the tumor that is present in that region. And you can use an external trigger. If it is superficial, you can use a laser light. If it is deep seated, you can use an X-ray trigger. And these nanoparticles can also act as radiosensitizers, and you can treat the tumor then and there. So there are two things that are achieved in this case. One is affordability, and the other thing is reducing the gap between diagnosis and treatment. And this material is an ideal theranostic material in the sense it could be used for diagnosis. At the same time, with an external trigger, you can make it into a therapeutic material and use it as a theranostic material. So all these work are possible mainly with collaborations. Okay, a single group is highly impossible to achieve all these things. Okay, so this kind of work we did in collaboration with IIT Bombay and Tata Cancer Center, which is in short called as ACTREC. And we utilized the concept. So when we started comparing with our earlier results, which lipose AU and the PLG AU, we understood that the rigidity of the material plays a very important role, and henceforth we started preparing materials which are a bit rigid, so that they do not lose their nano size, and we are able to achieve very good tumor targeting, and that is getting continued in many other works as well. Okay, and then we started using nano transformable gels, wherein this gel, when injected into one particular region, it starts to disintegrate in the form of nanoparticles, and this nanoparticle continuously gets into the circulation, and it even reaches those areas where the tumor has spread. Okay, currently for advanced stage tumors, that is something that has spread to many other areas, there is no specific treatment. When I say advanced stage, it is like it has got spread or it has metastasized to various other parts. Okay, so when it spreads to various other parts, there is only palliative care given to cancer patients. There is no proper cure or treatment given to them. So we thought this kind of material, because it is spreading to various other parts where the tumor is, we can use it for better treatment of advanced stage cancers. So we did few more studies, and then we were able to show that if you inject this material in one segment, it is able to go and reach the metastasized region. So here you see the lung nodules in which these materials have got accumulated. Generally, it is very hard to treat those tumors which have spread to other regions. So in this case, lung nodules. But now we have achieved certain positive results wherein this material is able to reach these metastasized lung nodules. Now, you might see that I have added one more question, treatment of advanced stage cancers, which is marked in dark, bold. Okay, my earlier question was only these two, that is affordability, degradability, whether selective drug delivery is possible, and then reducing the gap between identification treatment. Because at that stage, we were only having answers which is capable of addressing these questions. Now that we see that this material that we developed is capable of reaching even the metastasized tumor nodules, we can now start asking questions based on the results that we have already got. That is what I say, that there is a lot of conceptual reverse engineering that happens in research. Okay, so we didn't ask this question first because we were not having answers for it. Now that we have these answers, we started asking the right questions. So along with the earlier questions, we are now asking, can we have better materials for treating advanced stage tumors? So now another branch of research has emanated from these kind of positive results that we have got. Now, observation is very, very important in research. I'll give you a few examples which has led to certain publications, which has also led to certain grants for our group. Okay. So first thing is I'll speak about the observations that we had on liposomes and polymers. The other thing is a polymer related work, polyethylene glycol, and the third one is a startup and project related work. Now, here there were two types of groups which was working in my lab. One group was working on polymers, the other group was working on liposomes. If you see these two pictures, one is PNP, which stands for polymeric nanoparticles, the other is LNP, which stands for liposomal nanoparticles. Now, whenever we use polymeric nanoparticles, you see a diffuse red light coming in the cells. Whenever you use the liposomal nanoparticle, you see discrete red dots and red pigments coming in that region. Okay, the reason is there is a way by which things get delivered when you use a polymeric coating, and when you use a liposomal coating, the delivery differs. Okay, so when you use polymeric coating, there is diffuse uptake of these nanoparticles that happens in the cytoplasm, as a result of which you get diffuse red light. Okay, in the cytoplasmic region. Whereas when you use a liposome, the liposome goes and interacts with the cell membrane, and it attaches itself to the cell membrane, and as a result of which there is no diffuse uptake happening, there is only segmental uptake happening. So this is the novelty of this paper. Okay, so we were able to publish these kind of results in two different types of journals. Of course, we did various other studies, and that is because we started comparing these two things. Okay, so we had this data left over, and when we started comparing, we could see this difference very clearly. Okay, so that is why I say, like, observation is very, very important. And the other thing was an accidental finding in my lab, wherein we wanted to prepare nano shells and nano cages. So here you can see the nano shell, which is getting converted into a cage. In this, this is called as galvanic replacement of silver, wherein we use silver templates, and then we use chloroauric acid, and there is a redox reaction happening wherein the silver comes out and gets integrated with chlorine to form silver chloride, and the gold ion gets reduced to gold metal. And this is a very well-known process, and we use ethylene glycol for this kind of reaction. But the student did a simple mistake. Instead of taking ethylene glycol, the student took polyethylene glycol. But I must appreciate that she did not end up in getting these nanoshells or nanocages, but she came and told me that the material is fluorescing. Okay, that I have not heard, because we use PEG polyethylene glycol mainly for coating purpose, and we don't use it normally for fluorescent signals. But because this material was fluorescing, and she told me, like, I used the regular procedure, but then it was fluorescing. So I asked her to discuss with another student who was able to prepare these nanoshells, and then they found out that the mistake that was done was, instead of taking ethylene glycol, she took polyethylene glycol. Then we started understanding what are all the papers that got published with polyethylene glycol that is used for fluorescence, but we didn't get any single paper. The only few papers that we got was carbon dots that were made out of polyethylene glycol. Now, carbon dot is a completely different material. You see, this is carbon dots behave like quantum dots, wherein it emits fluorescence, and there are typical particles. Whereas you cannot use a carbon dot for its polymeric property, because everything has become carbon. Whereas there is an intermediate step in this process involved, wherein the fluorescent PEG not only retains the polymeric property of PEG, but it also starts to emit fluorescence, which are properties of carbon dot. So in this flowchart, you can see that from PEG, if you heat it, then there is an intermediate step involved, and then if you further heat it, it will become carbon dot. So there is an intermediate state wherein the PEG has properties of both the polymer as well as the carbon dot, and that kind of material now we are using it for coating as well as for tracking purpose using its fluorescence. And now we were able to change its fluorescence starting from cyan to green to yellow to reddish orange. Okay, so we were happy to get a couple of publications and one patent because of the student's observation. So I am thankful to the student for the mistakes that he made. So one humble suggestion that I would like to make is allow students to make mistakes, because we get patents, we get papers out of these mistakes. Coming to another segment, wherein we collected certain clinical samples, and we wanted to know if we could use it for diagnostic purpose. Again, this was some random idea. During my internship period in the medical college, there used to be a qualitative test that we people use for identifying cervical cancer. It's like they take acetic acid and they spread it on the cervical region, and if there is excess cancer cells present, it also has excess protein, and because of this acetic acid, the protein becomes white in color, and that gets the whitish region is considered as something related to cancer. So that was a qualitative test. So our idea was, can we somehow do with the help of nanotechnology to quantify the test? Okay, so we took samples, not clinical samples. In the beginning stages, we took samples which are having acetic acid in various concentrations, and we added gold precursors, and we were able to see that there is a typical color change involved. This has something to do with surface plasmon resonance and the size of the particle, and we see that there is a possibility of quantification based on colorimetry. And then we had to optimize the process. The typical step involved was in the amount of reactants that we use. If we use slightly larger amount of reactants, then there won't be any big difference. But then we had to spend some time in understanding what is the ideal optimal concentration that is required to cause this color difference. And once we were convinced, we started getting clinical samples. We collaborated with a hospital group which supplied us with the clinical samples, both the samples from the healthy patients and the cancerous patients, and we were able to develop a quantitative test for the identification of cervical cancer. And again, this was emanating from a simple idea wherein a color change could tell us the difference between a non-cancerous sample and a cancerous sample. We also did some analysis wherein we were able to see the lipos-AU kind of structure in the cancer sample, because we were already working with lipos-AU based on our prior experiments. We could easily correlate with this type of structure that happens with clinical samples, and this happened because of the excess cellular shredding, which had more amount of lipid in the sample, and still the study is ongoing. And we were able to get a BIRAC project from Department of Biotechnology, and we used it for having a startup. Okay, and every time when you have a challenge, you should look at it as an opportunity. I'll tell you an interesting thing here. Is it okay if I take some 10 more minutes? I've already it's already 12:30. Is it fine, sister? Okay, I'll continue. I hope I'm audible. Yeah. So here we wanted to register this company, and we thought we will name it as Infinite Health Tech. but we were told like this name is already taken, this infinite name. Okay, so this led to an opportunity wherein we were able to name our company as again infinite, but not using these same spelling. So instead of F5, we use the alpha, the numerical 5, and instead of TE, which ITE, instead of ITE, it spells as 8. So we use that again numerical 8. So this led to a name which is again pronounced as infinite, but if you look at the letters that are involved, it is called as IN5N8. Okay, and we were also able to create a new logo. So this logo will appear like a dumbbell, but you see this left hand side of the dumbbell, you see I here, and the right hand side of the dumbbell you see 8 here, and these two segments resemble N, and the center symbol is actually 5. Okay, so it reads as infinite. So this is a sweet opportunity that we got because of the issue that we faced, that our name has already been taken. And this test, we were able to give another typical name to it. It is like, you see the color change, so it is C color, which can be spelled as COL, standing for color, and the AU stands for gold-based reactions that are happening there. So we are making some baby steps. We do some incremental work wherein we added curcumin to the lipos-AU, and we use it for iontophoresis, and now we are venturing into antimicrobial resistance analysis as well. Of course, all these things are possible only through collaborations. Okay, so I will just give you some evidences wherein again observations are very important. When my student prepared this lipos-AU curcumin, they were able to see such small particles which were not zoomed, but we had a hunch that this could be the arrangement of nanocrystals in these liposomes. So again, analysis was done, and we were able to get the zoomed in picture wherein we were able to see the nanocrystals. But then there was a mechanism happening in situ wherein these nanocrystals were becoming microcrystals, and they were behaving as in situ depots to continuously deliver the drug. And that particular piece of work got published separately in a journal. That is because of our observation that though we prepare nanocrystals, but in cells we were able to see microcrystals, which means that there is a nano to micro transition that was happening at the cellular surface. And of course, the same material, because of the presence of gold, which is charged element, and a positive charge element, and because of the presence of curcumin, which can get activated by blue light, we used it for a dual light therapy. That is red light, which activates gold, and blue light, which activates the curcumin. And we used it for a combined photothermal and photodynamic treatment of acne. And I will skip these slides. Coming to the last slide, wherein we speak a lot about impact factor. My suggestion is that impact factors, they are just numbers, but we should also be aware that there is this San Francisco Declaration on Research Assessment, called as DORA, wherein the parties or organizations which are part of this DORA assignment, they don't rely on impact factors. They rely mainly on the quality of the work that gets published. Of course, impact factor is one of the factors, but that is not the main thing that decides their recruitment, or for award, or for anything else. Okay, so slowly the world is also moving away from these kind of number games. And some of the common possible mistakes that my own group has made, and which I would like to share with all of you, is that there will be a lot of spelling errors, there will be a lot of grammatical errors, sentence formation errors, etc. And we should ensure that whenever we submit an article, all these things are taken care, because a reviewer gets a lot of articles for review, and they are also faculty in various organizations, so they also have their own research work. So when they take up your paper, they should not get irritated. Okay, and unfortunately, after being in the academia for a longer time, only these errors come to our eyes. Okay, it's very easy to spot the errors. Okay, they will not be able to appreciate the good work that you have done, if at all they are able to see so many errors in your paper. So ensure that whenever you submit, even though if they are very small, but please take care and then submit it without any of these common errors. Okay. The other common mistakes that we do is that we abbreviate it according to our liking and want. So we had done, even for this fluorescent peg, we had made a mistake of naming it as MPEG, modified peg. But then the reviewer clearly pointed out that MPEG is not the right observation, right abbreviation for this, because there is methylated peg which is actually abbreviated as MPEG. Okay, so that causes a lot of confusion in the polymeric world. So he asked us to change it. Okay, he was a kind reviewer for us. And then as an early career researcher, I myself have made this mistake. I used to write the big projects in the beginning, but they were not very much successful, and I lost a lot of time and energy in those writing those big projects. So my suggestion is that at least during our early career stage, we should go for a couple of or 3 to 4 small projects, and then concentrate on publications rather than writing big projects. Big projects, of course, we should write, but after gaining a strong hold and after generating certain results, only then the committee will be happy to give you big funds. And when it comes to numbers, my suggestion, humble suggestion, is that you have at least 3 4 average number of projects, and concentrate more on your publications, even if it is 2 or 3 publications, but it should be in really high impact journals, so that you set the bar right in the beginning stage itself. If we start publishing in low impact or low quality journals, then that sets the bar, and we always tend to go for the easy way out. Okay, it is very challenging to publish in high impact journals, of course, I agree, but once you publish it, then you are setting a higher bar for your capability, and you will always strive to achieve those things. And other important thing as far as government grants are concerned is this utilization certificate and statement of expenditure. Both these things we'll have to submit on time. Only then the subsequent installments get released. Otherwise, there will be a big delay. And my strong recommendation is that if you are getting a 3-year project, you please apply for 6 months no cost extension or a 1 year no cost extension whenever your time is about to get completed. It is always easy to get a prior permission and then extend the project, rather than writing to them at the end stage where they also may not be able to help us. The current research focus of the lab has been in 4 areas. One is cancer nanotechnology. Of course, I have said a lot of things about it. Then the other is biomaterials and drug delivery. And recently, with the help of our partners, we were able to venture into antimicrobial resistance as well. So I'm thankful to all the funding agencies that has helped us to develop my lab. And I thank all the students of my lab, the collaborators, national, international, and the organizers of this program, Sister Alfonsa, as well as Dr. Jones Nadavar. Thank you. I'll be happy to take questions, if any.
Thank you, sir.
Thank you, Dr. Aravind Kumar. It was an excellent session. Most of the budding researchers are very good at analysis, experiments, field works, collaborations, and so on. The point at which they may get stuck is when they sit for a coherent writing. We used to say
Yes,
experience
ma'am.
is the best teacher. From an
I
MBBS
agree.
doctor to a biotechnology scientist, your experience is quite vast and wide that we could hear now actually. But this session has given us a right guidelines, framework, and tips one should follow in writing research articles. Thank you, Dr. Arvind Kumar. Now it's time to discussion. If anybody has any clarification or doubts, now I think we can take 5 minutes or more for just clarification. If anybody have any doubts or anything, you can just ask.
I think it was too much of an experience sharing for them. that they
Yeah,
are not able
maybe
to.
because of that. Even I myself feel actually we are also doing the almost little bit similar area, nanotechnology, nano fluid actually. So I think I can later contact you.
Sure,
That's
ma'am.
what I was
Sure,
thinking actually.
sure, sister.
Mm, I think there is no other doubts or anything. So once again, I express my deep sense of gratitude to Dr. Aravind Kumar for spending your valuable time with us. This session was very informative. Hope this will give confidence to the scholars and other researchers to write confident research proposals and articles. Thank you so
Yes.
much. Thank you.
Thank you, sister. I just want to share one small piece of information. It is like mostly I have shared my experiences and the mistakes that I made, and that will give definitely more confidence. There is no point in saying that I have achieved this and that. That is not going to give. We are all normal people, and everybody makes mistakes, and we learn from our mistakes. So when we used to study in school, I used to go very early to school, especially during exam period. That is mainly to see if other students also face the same problem that the overnight we used to study, but never used to cover all the portions for that particular exam. But when we reach school little early, then we see that every student has the same problem. Then we are satisfied. Yes. So we are
Sure,
not alone in this world. So everybody will
sure,
face
sure,
challenges. Okay. So we'll have to find out a solution for it.
sure,
Thank
sure,
you.
sir, sure. Thank you so much, sir. Thank you. So, on
Thank you. Thank you.
behalf of the entire college and the research fraternity, I extend our sincere gratitude, sir. Thank you.
Thank you, sister. Thank you all. Nice meeting you. I'll leave.
Thank you, Sister Alfonsa, and thank you, Aravind Kumar Regan, for this information and wonderful talk. Uh, Participants, please note that the feedback link is posted in the comment section. Please fill the Google form and make sure you have marked your attendance. The next session will be chaired by Dr. C.L. Joshi, and the invited talk will be by Dr. P.P. Pradyumnan. We'll begin the session by 1 p.m. and see you there. Thank you.